Hi, everybody. Who's here today? Give me a little hey in the chat so I can see who we've got for our lesson. Lesson 5 of our Year 12 summer school course. Hey, how you doing? Oh, I remember you're GCSE, right? But you're doing Year 12 as well. Is that right? Hey, Delight, how are you? Oh, I'm good, thank you. I'm good. Getting back into the swing of it for another day. Hey, good to have you here. So, what are we doing today? We are doing cell membrane structure, but we're also going to look at diffusion, which is a GCSE topic, but we're going to add on some A-level contents. Who's ready for results day on Thursday? Good question, Aria. Oh, yes, I saw your message on TikTok. I need to reply. I'll reply to you today, don't worry. I'm good. I had a really good weekend, thank you. I hope you guys were all good. Are you stressing about results day though? You're scared for results day. Oh, you'll be fine. You'll have to let me know on Thursday. I think we've got lessons today and Wednesday. So you'll have to message me and let me know on Thursday how you got on. What did I do this weekend? I went to a wedding, so it was really good. Went to a wedding on Saturday. Had a great time. There was a really good band. Don't worry. Right, okay. Should we make a start? Let's see what we've got. So, first thing I was going to ask you is just what you already know about the cell membrane. So, if you were asked, like, what is the job of the cell surface membrane or what is it made from? What do we already know about the cell membrane? Because we're literally just going to focus on that structure today. So, we've spent a lot of time talking about cells and organelles and all the different jobs. But at A level, you do a whole topic just on the cell membrane. Okay, so Esther says it controls what goes in and out of the cell. Yeah, Jawahir says controls what enters and leaves the cell. CS says it's made from cellulose in plants. CS, that would be the cell wall, not the cell membrane. Okay, the cell membrane is not made from cellulose. Sophia says it's made out of proteins. There are proteins in the cell membrane. You are right. Don't worry, CS. You don't really focus on what it's made from at GCSE, do you? You just focus on the function. So let's write down what we know. Controls what enters and leaves the cell. Can you give me any examples of ways in which things can enter and leave a cell? Any examples of the transport methods? Oh, Monkey says it's partially permeable. Yeah, let's write that down. It's partially permeable, which basically means some things can pass through it and other things can't. Right, good. So a few of you are saying osmosis can occur over the cell membrane, diffusion can occur over the cell membrane, and active transport can occur over the cell membrane. Perfect. So they were the 3 transport methods that you learned about at GCSE. And all of these things can take place across the cell membrane to help substances move in and out of the cell. That's basically it, right? That's probably all you learn at GCSE, I would say. Aria, share your news then. What was your news? Amazing news last week. Share it. I think we've got time. So A level then, we're going to look at the actual structure. CFK says, let me guess, there is more. There is more for sure. Yeah, we're going to look at actually the structure, what it's made from, and another type of transport method. In fact, we're going to do 2 more types of transport method at A level. Can I go back? Yeah, I mean, there was nothing too difficult on this slide, Aria. This was just stuff you probably already know, right? Just what the cell membrane does and then the different examples of transport across the cell membrane. You've already learnt those, so don't worry too much about that. Hey, M Wild. Okay, so what we're going to learn that's new then is the cell surface membrane, which at A level you can refer to as the plasma membrane if you want to, or you can just say cell surface membrane. It's made from phospholipids, okay? So these are a type of lipid. Just remind me, what is a lipid? What type of molecule is a lipid? Easy question, I'm sure. I'm just checking that our brains are still working. This is your first live lesson. Oh, nice. So you've watched the recorded lessons though, good. It's actually not a polymer. Good guess, but it's not a polymer because it's not actually made out of repeated monomers. It's not a protein either. Oh, I feel like it's a good job that I asked this question now. Yeah, it's a fat. Good. It's a type of lipid or fat. Okay, but fats actually aren't polymers. They're not made out of repeated units. They're not made out of repeated monomers. Okay, so it's made out of phospholipids and it forms a phospholipid bilayer. Does anyone know what a bilayer is? If you break the word down, bilayer, what does that mean? That prefix bi. Double layer, yeah, exactly. A double layer. A double layer or two layers. Yeah, two layers. Yeah, exactly. So this here, which you can see in purple and orange, this is a simplified drawing of a phospholipid. And over here, we've got a more complex drawing of a phospholipid, like you might see in chemistry. Now, you'll be pleased to know that in biology, you don't have to draw this one, okay? You don't have to be able to draw the full complex structure of a phospholipid, so don't panic. We just need to be able to represent it in this simple form. Okay, so this is the one that you would have to recognize and draw. Amazing news is that I'm getting my EHCP, hopefully 6 weeks. Oh, good. So you're going to get some extra support. That's good. Going to your dream sixth form. Well done, Aria. The picture on the left looks like a boxing glove. Yeah, it does a little bit, does it? But the reason we draw it like this is because we've got kind of two parts to this phospholipid. So we've got what we call the head, and then we've got what we call the tails. Okay. Now the head of the phospholipid is actually made out of two things. So the head is made out of glycerol. Hope you're writing this down. Glycerol and a phosphate group. And the tails, there are two tails, okay? And they are both fatty acids. Okay. So the head is the glycerol and the phosphate, and then we've got two fatty acid tails coming off of it. Now the head, we've described on my slide here as hydrophilic. Does anyone know what that means? Hydrophilic. Whereas the fatty acid tails are hydrophobic. What do those key terms mean? Because you do have to be able to define them in A-level biology. Esther says water loving. Yeah, something to do with water. Yeah, water loving. And water hating, yeah. Now, we're not going to say that in an exam, but you are right. Yeah, that's kind of what it translates to, water loving and water hating. But hydrophilic is the water loving bit. It's basically attracted to water. Whereas hydrophobic, the water hating bit, as you guys have said, means it repels water. Okay? Yeah, so it kind of makes sense. So the fatty acid tails repel water, whereas the head, which don't forget is your glycerol and your phosphate group, they are going to be attracted to water. Now let's just quickly look at the complex phospholipid. I've already said don't worry about ever drawing this, but I'm just going to label the group so you know what you're looking at. So this here, this is the phosphate group. okay? This here, ooh, got a bit wonky there. This is the glycerol, okay? And then you can see over here, sticking out, we've got our 2 fatty acids, okay? So the glycerol and the phosphate group make the head, the water-loving or the hydrophilic head, and then the 2 fatty acids make the tails. Is that why oils and fats don't mix well with water? Yeah, because of that hydrophilic head. Sorry, said it the wrong way around there. Because of the hydrophobic tails, yeah, they're repelling water, aren't they? So you're spot on. Fats don't mix well with water. And you'll see that even when you're doing the washing up and you've got like oil or fats on your baking tray or whatever in your frying pan, those hydrophobic fatty acid tails repel the water. Does the glycerol include the oxygen? That oxygen, yeah, it was part of the glycerol, but can you see here, this is now forming a bond. So these, I'll just triple underline them. These oxygens were part of the glycerol originally, but now they're being used to form a bond between the glycerol and the fatty acids. These bonds are actually called ester bonds. I mean, I wasn't going to go too deep into this today, but we have got 2 ester bonds between the glycerols and the fatty acids. Can you still get this complex phospholipid? Yeah, they might give it you CFK. They might give you a diagram like that in an exam, but you would never be asked to draw it, is what I was saying. So don't worry about learning it off by heart, but you should be able to recognize the parts if they gave it to you in an exam. So the phosphate group, the glycerol, you might have to label them or draw a box around them or something. You might have to name the bonds, which are called ester bonds. You might have to label the fatty acids. Yeah, you probably do ester bonds in chemistry. What's psychology like, Laura? Yeah, I did do psychology. I actually really enjoyed psychology. Do you know, fun fact, I actually taught psychology A-level for 2 years, right at the start of my teaching career. I wasn't meant to. I was qualified to teach biology, right? But when I started teaching, they were short of an A-level teacher and they couldn't find an A-level psychology teacher. So they asked me if I'd be willing to give it a go. And I did. And I actually taught A-level psychology for 2 years because there's lots of biology in it. And I did enjoy it. Okay. You chose chemistry even though I didn't do it for GCSE. Surely you had to do chemistry for GCSE. Even if you do combined science, you have to do chemistry. Did you ask for a pay rise? I didn't get a pay rise. Didn't get any extra money. No. Okay, right. So let's have a look at the cell surface membrane then. This is what you're going to see at A-level. So it looks a little bit different to GCSE. Before we look at this in too much detail, can we just actually check we know which bit is the phospholipid? Let me just put a box around one phospholipid. Okay, so this here, can you see where you've got this? They've really simplified it. But this is one of those phospholipids that we've just been discussing. Okay, I'll just do another one for you. So can you see here? That's a phospholipid. That's a phospholipid. Okay, so we've got what we call a phospholipid bilayer. We've got 2 layers of phospholipids. And if I do you a really simplified picture of a bilayer, a phospholipid bilayer, let me just do a really simple one. Can you see it's like this? So these are all phospholipids. We've got the head, and then inside we've got the tails. But that's what we call a phospholipid bilayer, and that's the main part of the cell surface membrane. What are the tiny circles next to those phospholipids? So, do you mean these things here? This is cholesterol. Let me just draw another one here. Cholesterol. So there is cholesterol between the fatty acid tails. Okay, so you've got your phospholipid bilayer, and then if I draw some cholesterol on mine, you've got cholesterol. Did we know that? Did we know there was cholesterol in a cell surface membrane? Because you always think of cholesterol as a bad thing, right? You think, oh, if you have high cholesterol, it's going to lead to heart disease and heart attack. But we actually need cholesterol in our diet to make our cell membranes. And our liver makes cholesterol as well because we do need it. It is a key component of the cell surface membrane. So you've got your phospholipid bilayer, which is the 2 layers of phospholipids. And in between those phospholipids, you do have cholesterol. Yeah, there's high density lipoprotein and low density lipoprotein. Yeah, that is what it's called. Okay, so I want you to answer me a question then. Why do you think, based on the properties of these phospholipids, based on the hydrophilic and hydrophobic nature of the heads and tails, why do you think they arrange themselves like this? Why have we got the heads on the outside and the tails on the inside? We've got the heads on the outside and the tails on the inside. Why do you think that is? Thinking about the properties of how these substances react with water. Yeah, the cholesterol is between the tails. So we've got a few answers here. The heads are on the outside, so water can enter. Water can enter, you are correct. Yeah, water can get through. The heads are hydrophilic, so water can dissolve via osmosis. Yeah, kind of right. Yeah, it helps water to pass through. Okay, let me just explain. What you might not realize is there is water outside the cell. And there is also water inside the cell. So if this is outside and this is inside, there is water on both sides. There's water inside the cell in the cytoplasm. The cytoplasm is mainly water, okay? So cells are full of water inside, but also on the outside of the cell, there's something called tissue fluid, which is mainly water. So all cells are kind of surrounded by water, both inside and outside, okay? We'll go through the other bits in a minute, don't worry, monkey. So that's why they have to arrange themselves in this way, okay? So I'll do you a little written description. What we'd say is the heads are on the outside because they attract water. And there is water outside the cell on both sides, outside the cell and inside the cell. So the heads are on the outsides of the bilayer because they attract water. And the tails are inside the bilayer because they repel water. It's all to do with water. Because there's water on both sides, the heads are going to be attracted to that water, so they end up going to the outsides. The tails are repelling that water, so they end up going to the insides. Okay, but yeah, it does help to control. Water can get through this. We know that from GCSE. But the way in which these phospholipids organize themselves is really because of those hydrophobic and hydrophilic properties, the heads versus the tails. The heads attract, the tails repel, so they end up in this bilayer formation. The tails are kind of protected on the inside away from the water, okay? Now, a lot of you have been asking me what are all the other parts, because so far we've only labelled the phospholipids and the cholesterol. So these big circles are basically proteins, okay? You see all these big white circles. I'm going to run out of space. These are all proteins embedded in the phospholipid bilayer. And we've got lots of different types of proteins. So some of you have said, like, what are the squiggly things? What are the round things? Let me find a good example. So it's so hard to figure out what's going on. This big one here that I've just labeled in the center, this is what we call a glycoprotein. Okay. Yeah, Esther, well done. I can tell it's a glycoprotein. You've got to look really carefully, but I can see this big protein. This is a big protein here. But can you see attached to it, you've also got a chain, a carbohydrate chain. I hope you're following my pen. So you've got a big protein with a carbohydrate chain attached. So that would be a glycoprotein. And then if you look at the squiggly wiggly thing here, this is actually a glycolipid, this one. That's not a protein. You've got a squiggly wiggly lipid, and then you've got some carbohydrate chains attached to it. So you've got all sorts going on. Now, we're not going to spend too long going through all the detail of everything, but what I want you to take away is the structure, okay? So you've got your phospholipid bilayer, which is key terminology. Phospholipid bilayer. We've also got cholesterol. We've also got some glycoproteins, carb plus protein. We've got some glycolipids, yeah, carb plus lipid. And these are all kind of embedded or dotted throughout the phospholipid bilayer. So is the large empty circle a glycoprotein? Yeah, it's not really an empty circle. That big white circle is a big protein that spans the whole bilayer. That's a massive protein. And then it's got a chain of sugars, a carbohydrate chain attached to it. But it's not really an empty circle. That is a protein. You can see another protein here, this big white. But this is another example of a glycoprotein because I can see some carb chains attached to that protein. So that's another glycoprotein. Okay, we're going to come back to this later. Okay, we're going to label another one of these. So the proteins remain in the membrane. Yes, they are part of the membrane structure. Glycoproteins often act as antigens. They're used for recognition of the cell, and they can act as receptors for hormones. They have all sorts of different roles in terms of like cell recognition, cell signaling, cell communication. They have all different jobs, okay? Let's see if you can answer this for me without me telling you. At A level, we say that the cell membrane is like a fluid mosaic. We call it the fluid mosaic model. You know, like how we talk about enzymes and we say the lock and key model. Well, when we talk about membranes, we describe it as the fluid mosaic model. Anyone got any ideas why? Why would we describe it as fluid? What do you think that means? And why would we describe it as looking like a mosaic? What do you think that means? The fluid mosaic model of membrane structure. So Jawar here says it can flow. Yeah. There is movement there. Yes, good. The phospholipids are fluid. It's not rigid. Good. Not rigid like a cell wall. Patches up, kind of. The proteins inside are the mosaic. Yeah. Yeah, good. I would say fluid. That's to do with the phospholipids. So the phospholipids can move. Now, I'm not talking like loads of movement. They're not going to get up and move away, but they kind of move like this. They can move from side to side. I'm going to put move slightly from side to side. So they don't swap places or get up and move around, but the phospholipids can go like this. They can move from side to side. So there is fluidity and there is movement. Someone said it's not rigid. That was quite good. I like that. The membrane is not rigid. Can you think of a factor? I'm just going to push you a little bit further. Can you think of a factor, using what you know about maybe chemistry, something that might make the membrane become more fluid and therefore more permeable? Because if the membrane does become more fluid, if the phospholipids start moving more, it becomes more permeable. Now, what factors do we know about that kind of cause, give more kinetic energy? Let's think of it that way, because it's not water. So what would give those phospholipids more kinetic energy? Temperature, heat, right? Yeah. So an increase in temperature increases the fluidity. And that does make the membrane more permeable. You're going to do a practical on this. In year 12, you'll take some cells. We usually use beetroot cells, and you put them at different temperatures, and the membrane becomes more and more fluid and more permeable. And then more of the pigment, the red pigment in a beetroot, leaks out through the membrane. You can actually measure it. But yeah, increase in temperature does make the membrane more fluid. And equally, a decrease in temperature makes the phospholipids less fluid, and it makes the membrane less permeable. What exam board do I teach? AQA, but also OCR. Mosaic, someone's already said this, it looks like a mosaic. We all know what a mosaic is, right? Because proteins are like embedded in the phospholipid bilayer. So you've got, if I just do another quick picture. So you've got your phospholipid bilayer. Let me just rub that one out actually. You've got your phospholipid bilayer. Do my little fatty acid tails. Remember, these are 2 fatty acid tails. But it looks like a mosaic because within that phospholipid bilayer, you've got these kind of proteins that are kind of stuck in there, right? You've also got cholesterol. I'll just do some little bits of cholesterol in between the fatty acid tails. But the proteins look like a mosaic. Okay. I just want to give you another piece of vocab, if you don't mind, if you're kind of comfortable. Some of the proteins are what we call intrinsic proteins, and some of them are what we call extrinsic proteins. Now, based on how I've drawn them on my diagram, does anyone think they know what the difference is between an intrinsic and an extrinsic protein? Or sometimes these ones can be called peripheral proteins. That might give you more of a clue. Yeah, good, Zainab. So we can differentiate between them. Yeah, intrinsic because it goes through both layers. That's really good. So intrinsic proteins go through both layers of phospholipids. They basically, another way of saying it would be like they span the whole bilayer. They go from one side to the other. Whereas extrinsic proteins or peripheral proteins are only on one side of the bilayer. Yeah, so you might see that as well. You might see that come up. Now, no, I'm not going to ask that yet. That's too hard. Okay, are we okay so far? Now, we're not going into too much depth here because I am just focusing on structure. But have we got this basic idea? We've got our phospholipid bilayer, so 2 layers of phospholipids. We've got cholesterol. We've also got lots of different proteins. We can have proteins that span the whole bilayer, proteins on one side of the bilayer. We have glycoproteins. We also have glycolipids. So it's a lot more complex than what you've done at GCSE. Okay, perfect. We're going to have a go right at the end. We're going to label a full membrane. So we'll see what you can remember at the end. Perfect. Okay. So the other thing that comes up in this topic, obviously, as well as the structure, we have to talk again about transport methods. So you're going to be doing diffusion, osmosis, and active transport again at A level, as well as a couple of new methods. So I thought we'd just start with the most basic type of diffusion. Now at A level, this is where you're going to start to hate me. There are different types of diffusion. Okay. Don't know if you know that yet, but there are. So the one that you did at GCSE, we're now going to call it simple diffusion. Osmosis is a type of diffusion, yes. Diffusion of water, but we're also going to learn about another type. Okay. But simple diffusion is basically the one you did at GCSE. So if I were to say to you, what is diffusion? What is simple diffusion? What would be your GCSE definition? Simple diffusion. What do you reckon? Who can remember their best GCSE definition? And this is just what we call at A-level simple diffusion. So Esther says the movement of particles from an area of high concentration to an area of low concentration. That is perfect. Or the movement of molecules is fine. Molecules or particles or even a substance is fine. And you don't have to say anything else at A level, okay? It's just the movement of particles from an area of higher concentration to an area of lower concentration. And we're going to say exactly the same at A level. So you're absolutely, yeah, the net movement, that's a really good way of saying it, the net movement, because particles are moving in both directions, right? But overall, there's a net movement from a higher concentration to a lower concentration. So, for example, I don't know what this is. Should we say this is oxygen? These little triangles. So if we're going to imagine this is oxygen, oxygen can just simply diffuse. It's going to go from a higher concentration to a lower concentration. Talia says, is it over a concentration gradient? What we say, Talia, is it's the movement down the concentration gradient, not over. We'd say it's down the concentration gradient, down because you're going from high to low, right? So you're going down the concentration gradient. So simple diffusion, let me show you how it would work. It would simply diffuse straight through the phospholipid bilayer. So simple diffusion occurs through the phospholipid bilayer. Okay, straight through. Doesn't involve a protein. It goes through the phospholipids past the fatty acid tails. Okay. Are there glycolipids in this diagram? In this diagram, no. All I can see on this diagram are phospholipids. I can see an intrinsic protein, which is purple, and I can see some extrinsic proteins, which are orange, but I can't see any glycolipids or glycoproteins. Can't see any cholesterol here either, but it would be there, right? This is just simplified. So simple diffusion occurs through the phospholipid bilayer. Now we need to start thinking what substances can do this. Oxygen and carbon dioxide. Oh, and water. But remember, when it's water, we do call it osmosis, but osmosis is a type of diffusion. Osmosis is just the simple diffusion of water. We just call it osmosis when it's water, but it is diffusion, okay? So these substances, things like oxygen, carbon dioxide, and water, they can just simply diffuse through the phospholipid bilayer. Why? It's not waterphilic, it's because these substances are lipid soluble. So basically, they can dissolve in the fatty acid tails. They are soluble in lipid. Oh, you're going to ask me a question now, aren't you? Are you going to ask me a question about the water? Water is an exception. It's not actually lipid soluble. Was anyone on to me? Was anyone already thinking, hang on a minute? Water and lipids do not mix. We said that before. They do not mix, water and lipids. But water is an exception to this rule. Because water is so small, it can simply diffuse, okay? It is so small that even though the water is not lipid soluble and it doesn't mix well with lipids, it's so small that it can get through the phospholipid bilayer by simple diffusion, okay? And then oxygen and carbon dioxide, they are really small, they are lipid soluble, they can simple diffusion, simply diffuse through the phospholipid bilayer. Okay, but water is an odd one. I'm going to give you that. It's an odd one. You don't expect it to be able to diffuse because water and lipids do not mix. It's not lipid soluble, but it's so small that it can get through. It's quite slow. It's not quick. It's quite slow, but it can diffuse through the phospholipid bilayer. But if you can for me, try and remember a couple of examples. Okay, so oxygen and carbon dioxide are the easiest ones to remember. They are lipid soluble. They can use simple diffusion. How do they get past the phospholipid heads? Again, it's simply because they are so small and because, remember, they're going down a concentration gradient. There's such a high concentration of oxygen, for example, outside the cell, and it's so small that it can move between the phospholipid heads and then it dissolves through the bilayer and diffuses from a high concentration to a low concentration. So it's to do with the fact that it's lipid soluble, it's small enough, it can kind of sneak between the phospholipids. Okay, so that's simple diffusion. On this diagram, what are the two purple sticks? Good question. We're going to look at that on the next slide. So it's a protein. It's an intrinsic protein because it spans the whole width. But this specific example is what we call a channel protein because it's formed a channel in the membrane. So it's still a protein. It's still an example of an intrinsic protein. It spans the whole bilayer, but that specifically, because it's formed like a pore or a channel, it's called a channel protein. Yeah, the orange ovals are also proteins. These would be extrinsic proteins because they're only on one side of the bilayer. Obviously, they are not channel proteins. They haven't formed a channel. They are just extrinsic proteins. Okay. So simple diffusion, let's not complicate it. It's the same as GCSE. It's the movement of particles from a higher to a low concentration. All we're saying at A level is that simple diffusion, they diffuse through the phospholipid bilayer, and it's things like oxygen, carbon dioxide, and water. So let's have a look at a different type of diffusion. Facilitated diffusion. Now, guys, if you think of the word facilitated, to facilitate, what does that mean in the English language? Because that's exactly what this is. Facilitated diffusion, or to facilitate diffusion. To allow, it's even a little bit more than allow. I think, like if I said to you, your teacher facilitates your learning, I facilitate your learning. What am I kind of doing? I'm making it a little bit easier. I'm assisting. I'm helping. Exactly. That's kind of how I think of facilitated diffusion. It's diffusion with help. Now, obviously, I'm not going to say that in an exam, okay? but it's diffusion. Substances that can't quite diffuse on their own need a little bit of help to get across the membrane. Yeah, it makes it easier. Exactly. So some substances cannot use simple diffusion. This is because maybe they are just too big. They can't get past the phospholipids. Maybe they are not lipid soluble, so they can't dissolve in the phospholipid bilayer. Maybe it's because they are charged or polar. Who's heard of that word before? Polar. Polar. For biology, I don't know whether you do this in chemistry and you might go into more detail, but for biology, we can just think of it as like they have a charge. Yeah, they have a charge. Like the North and South poles on a magnet. Yeah, they have a charge. So if something is too big or it's got charges or it's not lipid soluble, it's not going to be able to use simple diffusion. Yes, Zainab, I knew someone was going to know this. So remember, water is the exception to this rule. Zainab has just made a really good comment in the chat. He said, water is polar. Yes, it is. Because the hydrogens have a slightly, we call it delta positive, and the oxygens are slightly negatively charged, delta negative. Water is polar. So it shouldn't really be able to use simple diffusion because it doesn't mix well with lipids and it is polar, but water is the exception. Okay, it can use simple diffusion. Yeah, let's just leave it at that for now. Water is such an odd one. I don't want to throw too much at you in one lesson, but water uses both. Water uses simple diffusion and it can use facilitated diffusion. Because even though it can use simple diffusion, it is quite slow. So it also uses facilitated diffusion. Yeah, it's so small. Think of it like that. It's so small that it can use simple diffusion, but cells also move water with facilitated diffusion to speed it up, especially cells like the cells in your kidneys that need to move water quickly. They will use a lot of facilitated diffusion for water because simple diffusion is too slow for water. So facilitated diffusion, let's put a definition down. It's exactly the same as simple to begin with. So it's the movement of particles from an area of, it's still the same. So we're still going from an area of higher concentration to an area of lower concentration, but it uses a transport protein. That's the difference. It needs a transport protein. It can't just move through the phospholipid bilayer. It needs a protein. Okay, so this is where the proteins come into play. So examples of substances that use facilitated diffusion. So things like glucose and amino acids, because they're quite big. I'm just going to put quite large, okay? They can't use simple diffusion because they're too big. It's not active transport, okay? Guys, how is this different to active transport? Active transport is very different. They both use proteins, but this is a type of diffusion. It's not the same as active transport. It's passive. It is passive. Active transport uses energy, yeah. And this is from an area of high to low. That's the big difference. Active transport moves substances against the concentration gradient from low to high, and it requires energy to do so. This is still diffusion. I'm so glad you asked that question. This is still diffusion because we're still going from high to low and it is still passive. I'm going to just put here, it's still passive, guys. It doesn't require energy and it's still going down the concentration gradient. Okay, so that's the difference. So glucose and amino acids, ions, e.g., I don't know, potassium ions, sodium ions. What other ions do we do? Calcium ions, is that Ca2 plus? Any kind of charged ion. Water can use a facilitated as well because it's polar. Okay, so if it's too big or it's charged, it's polar. When we talk about this, we quite often talk about ions. Yeah, calcium ions, sodium ions, potassium ions. What other ones do we talk about? Just thinking what magnesium ions. All of these things need a protein. Okay, water is a bit of an odd one. It can use facilitated diffusion and it can use simple diffusion. It's a bit of an odd one. It is polar, so it does have a charge. It has two opposite charges. Yeah, polar is basically charged. Water has like two opposite charges. It has the slight positivity of the hydrogen and the slight negativity of the oxygen. Okay. Would a random element use simple diffusion? Like oxygen and CO2, I suppose we don't really ever talk about just like a random element. In biology, it's always going to be oxygen or carbon dioxide, or we're going to have glucose and amino acids. We're going to have ions. There's never just like zinc. I don't know. Okay. Same in plants, yeah. This is plants and animal cells. Same in plants now. The structure of the cell membrane is the same in plant cells and animal cells. Okay, so let's have a look at facilitated diffusion. So facilitated diffusion uses two types of protein. I'm going to put two types, I hope this is okay. I'm going to put two types of intrinsic protein. Remember, intrinsic proteins span the whole bilayer. And that makes sense. If you want to move something from outside the cell inside the cell using a protein, the protein is going to have to span the whole bilayer. It's going to have to be an intrinsic protein. It's going to have to cross the whole bilayer. Otherwise, you're not going to get the substance across, right? So it uses two types. These are intrinsic proteins. Oh, I've already written it, guys. They span the whole width of the phospholipid bilayer. Are transfer RNAs carrier proteins? No, because they're not in the cell membrane. They're not made of protein. They're made of RNA. They're made of nucleic acid. They're in the cytoplasm. They go to the ribosomes. They carry amino acids to the ribosome. So no, they're not part of the cell membrane. They're not made of protein. So a channel protein is basically a channel or a pore in the cell membrane. It basically just, it's like an alleyway. Can you imagine if you're trying to get from one street to another, but there's loads of buildings in the way and you can't get through? A channel protein is literally like an alleyway. It allows substances through the channel. So these substances might be ions, they're charged, they might be too large, it might be glucose, it might be amino acids. They cannot, let me do a little arrow, they cannot diffuse through the phospholipid bilayer. They can't do that, okay? So instead, they're going through a little channel that's provided for them, okay? Easy. It's facilitating diffusion. It's allowing them through. This is why we say the membrane is partially permeable, right? Some things can pass through the phospholipids by simple diffusion. Other things need a channel protein. Some things can't get through at all. So that would be a channel protein. Carrier proteins are a little bit different because the way a carrier protein works, it's not so much a pore, it's the substance binds to the protein and the protein changes shape. To move the substance across. So it's not so much like an alleyway. It's like, what could we compare this to? It's like someone picking you up and physically moving you across. Does that make sense? You can see it on the diagram. So whatever it is that's moving binds to the protein on one side of the membrane, That causes the protein to change shape, which delivers the substance to the other side of the membrane. Okay, it's not a type of enzyme, no, because to be classed as an enzyme, you have to catalyze a chemical reaction. You have to convert a substrate to a product. You have to change it. This is not changing it, it's just moving it. It's not reacting with the molecule. It's not creating any kind of product. It's just moving it across. Okay. But look, guys, both of these examples, we are going from high to low concentration. Okay. The only difference between this and simple diffusion is this is using a transport protein, e.g. a channel or a carrier protein. Okay, that's it. What's an E. coli? E. coli is a type of bacterium that causes food poisoning. The green circles look like they are engulfed. They're not really engulfed. It's more like, let me do it. Here's my carrier protein. So it binds to the carrier protein and that causes the protein to change shape. And when it changes shape, I can't draw it, I'm so rubbish. When it changes shape, it can then get through. It moves it to the other side. That's really hard. I think the diagram probably shows it better than I have. So yeah, it just binds to it and the protein changes shape. Channel protein, there's nothing really to explain. It just creates a channel in the membrane. That's it. It's just a pore. It just creates like an alleyway in the membrane. So we just say it's a protein, it's a channel protein. It creates a channel in the membrane. Yeah. I'm not sure if it's really soft so it squeezes through. It's more about, Zainab, the protein changes shape. That's how I would describe it. I'm trying to think of an analogy, but I can't think of one. But it changes shape. The protein changes shape. Okay. Let's have a look at a past paper question then. Yeah, we're going to finish on time, don't worry. This is quite often how they test you. So it's not always easy because they quite often use graphs. Okay. The thing is on the diagram, there is no other opening on the, oh, there isn't. Yeah, look. So it binds. So this is like step 1, Talia. It binds, and when it binds, the protein changes shape. This is like step 2. So this is kind of, I think this is meant to be showing you like this is the same protein. So this is the protein to begin with, and the substance binds in step 1. And then when the substance binds, it changes shape. So now it looks like step 2, I think. I think that's how the diagram works. So the rate of both types of diffusion is increased if the concentration gradient is steeper. We know this from GCSE. But now we're learning about facilitated diffusion. So the rate of facilitated diffusion can be limited by the number of channel or carrier proteins in the cell surface membrane. Okay, so we know with any type of diffusion, the steeper the concentration gradient, the faster the rate of diffusion. We know that from GCSE. But now with facilitated diffusion, the rate will be limited. It will be restricted by the number of transport proteins. Yeah. Using this information, which substance shown on the graph uses simple diffusion? We're going to label the membrane to finish with ARIA right at the end. So we've got 2 substances on this graph. We've got a monosaccharide, which is a simple sugar. And we've got a monoglyceride, which is a simple lipid or fat. Now, which one do we think uses simple diffusion? You need to look at what's on the x-axis. On the x-axis, we've got the external concentration of the molecule. So as we go along the x-axis, we are increasing the outside concentration of the substance. And then we've got the rate of uptake. Good. You're giving me the right answers. I just want to know if you actually understand it. Yeah. Which substance shown on the graph uses simple diffusion? It is the monoglyceride. Now, how did we know that? The monoglyceride, this green line, what it's showing us is if we increase the external concentration, the rate will keep increasing. Because as we increase the external concentration, we're basically increasing the concentration gradient. If we keep adding more and more and more and more of this substance outside the cell, it will keep going in faster and faster and faster because it's using simple diffusion. But which substance uses facilitated diffusion? That has to be the monosaccharide. Can you see how it plateaus? It gets faster to begin with, but something is limiting the rate of uptake. Okay, so explain the change in the rate of uptake for the monosaccharide, which is the purple line, between the points marked A and B. So explain. I was going to describe it then. I was going to describe it and say, as you increase the external concentration, the rate of uptake increases. But that would be described, wouldn't it? How are we going to explain it? As we increase the external concentration, I'm going to explain it. The concentration gradient gets steeper, which increases the rate of diffusion. And at this point, the number of transport proteins is not limiting. Does that make sense? So to begin with, remember we're looking at A to B here. Between A to B, as you increase the external concentration of the monosaccharide, you're going to increase the rate of diffusion because the concentration gradient is going to be getting steeper. And at this point, the number of transport proteins is not limiting the rate. Exactly. They're not a limiting factor at that point. But between C and D, the number of transport proteins is limiting the rate of diffusion because this is facilitated diffusion. I'm going to put that down. This is facilitated diffusion. This monosaccharide needs a transport protein to get into the cell. And there's only so many of them in the cell membrane. That's what I want you to understand. So it's going to be limited by the number of transport proteins, whether that's channel or carrier proteins. There's only so many. So it's reached, by the point time you get to C, it's reached a maximum rate. Can't move in any faster. All the transport proteins are being used. How did I know the monoglyceride uses simple diffusion? Because if you look at the green line, Talia, the rate just keeps going up. It's directly proportional. If you increase the concentration of the substance outside the cell, the rate will just get faster and faster. It's not limited by anything. It must not be using a channel protein or a carrier protein because there's nothing limiting the rate, if that makes sense. So the steeper the concentration gradient, the faster the rate of diffusion. Whereas for the purple line, it levels off. Something is limiting the rate. That's how we know it must be facilitated diffusion. It must be relying on a transport protein. Okay. Does that make sense, Talia? It's like saying you can increase the external concentration, but it can only go so fast because there's only so many transport proteins in that cell membrane. So no matter how much substance you add to the external environment, it can only move it in so quickly. It will reach a maximum rate. Okay. Okay. Right, we've got a little activity quickly to finish. This is only going to take us 1 minute, so hopefully we won't go over. by too much. I want you to tell me what the labels are. This is a full diagram now of a cell surface membrane. I've tried to include everything that we've discussed on this diagram. So what I want you to do to finish is in the chat, put the letters A, B, C, D, E, F. Have I got everything there? And give me your labels. And I'm going to wait until I see all the answers, I think. And then I'll put the correct ones on. Good, Zainab. Good, Mary. Keep going. C and F, you might forget, I suppose. Have a little think carefully about C and F, because you've got two things joined together for C and F. Good. Good, Zainab. Good, Esther. Good, Mary. C is there is carbohydrate there, but what's it attached to? And then what do we call that if we've got carb and lipid together? Can you remember? B is the phospholipid. It means specifically it's pointing to the head. So we could just say phospholipid head or phospholipid. C, yes, C is a glycolipid. Not often asked about these, to be honest, but I can see there's meant to be a carbohydrate chain and it's attached to a phospholipid. So we've got part carb, glyco, and part lipid, glycolipid. F, good, is a glycoprotein because I've got part carb, glyco, part protein, which is the big blue circle. So that would be a glycoprotein. Good. E, channel. So yeah, there's a few things we could say for E. We could say it's an intrinsic protein. It looks like a channel protein, doesn't it? Kind of. So maybe that's meant to be a channel protein. Maybe that's a protein that is used for facilitated diffusion, right? Or it could be a carrier. Yeah, it looks like the channel's not open at that end. I think it was meant to be a channel protein, I'm not going to lie. But I think the drawing is maybe just a little bit dodgy because it kind of looks like it's closed at one end. So maybe we should say it's a carrier protein that's going to change shape. And D, does anyone know what D is? Has anyone got D? Can't actually see the correct answer for D. So D is a small molecule. It sits between the phospholipid tails. Good, Zainab, it's cholesterol. Okay, so we've got, I want to do a big bracket here as well. Phospholipid bilayer with the proteins embedded in it. We've got intrinsic and extrinsic proteins. We've got some glycoproteins. We've got some glycolipids. We've got cholesterol. So this is kind of what I wanted you to be able to do by the end of the lesson. But also don't forget what we've learned about simple diffusion. Simple diffusion is where substances diffuse through the phospholipid bilayer. But we've also got facilitated diffusion, which uses a transport protein. Okay, we have finished. We're pretty much on time. Not too bad. Okay, thank you for joining me today. I hope you found it useful, especially I know some of you, it was your first live lesson. So thank you for joining us live. I hope you can do the same on Wednesday. And make sure you give your homework a go, and I will see you next time. Enjoy, guys. Enjoy your other lessons. I'll see you on Wednesday. Bye, guys. Thank you. Bye.